The development of the plaque assay was a key to the blossoming of the area of virus genetics, much as Drosophila was to eukaryotic genetics. The plaque assay offers several advantages. For instance, you can precisely enumerate the progeny. You get production of pure clonal strains, and you can assay for conditional lethals. Now with electrophoresis, drug treatments, molecular methods, and so on, we can assign properties to viral genes and identify viral gene products. Viruses are continuously changing as a result of genetic selection. They undergo subtle genetic changes through mutation and major genetic changes through recombination. Mutation occurs when an error is incorporated into the viral genome. Recombination occurs when co-infecting viruses exchange genetic information, creating a novel virus. We'll talk about both of these processes in this lecture. The mutation rate of an organism is defined as the probability that a change in genetic information is passed to the next generation. In viruses, a generation is often defined as a cell infection cycle, which includes attachment to the cell surface, entry, gene expression, replication, encapsulation, and release of infectious particles. Mutations are not restricted to replication, since they can also result from editing of the genetic material or spontaneous nucleic acid damage. The remarkable capacity of some viruses to adapt to new hosts and environments is highly dependent on their ability to generate de novo diversity in a short period of time. Rates of spontaneous mutation vary considerably among viruses. For instance, RNA viruses mutate faster than DNA viruses. Single-stranded viruses mutate faster than double-stranded viruses. And genome size appears to correlate negatively with mutation rate. Viral mutation rates are modulated at different levels, including polymerase fidelity, sequence context, template secondary structure, cellular microenvironment, replication mechanisms, proofreading, and access to post-replicative repair. Our current knowledge of viral mutation rates indicate that viral genetic diversity is determined by multiple virus and host-dependent processes, and that viral mutation rates can evolve in response to specific selective pressures. Knowledge of the processes underlying viral mutation rates has implications for understanding and managing drug resistance, immune escape, vaccination, pathogenesis, and the emergence of new diseases. In clinics, the importance of viral mutation rates can be illustrated by the history of anti-HIV treatment. The nucleoside analog AZT was the first approved anti-HIV drug, but unfortunately, the appearance of drug-resistant variants rapidly frustrated this monotherapy. HIV-1 is a fast-mutating virus and produces every possible single-base substitution, including the ones that cause AZT resistance, within a patient every day. The subsequent success of highly active antiretroviral therapy, or HAART, did not reside on merely increasing drug potency, but mainly in combining different groups of drugs, including AZT, such that the chances of resistance mutations appearing were minimized. Now, we have to consider some terms. Wild type is an arbitrary designation. The wild type is the original strain from which mutants were selected. It is usually lab-adapted and may not reflect the nature of the virus or the gene as you would find it in nature. A field isolate is a new isolate from a natural host. Unfortunately, there is no universally accepted definition for the terms strain, variant, and isolate in the virology community. It is important to point out that no matter at which classification level one examines a virus, one always deals with a varied population. A virus-infected cell will, after only one round of replication, already contain a population of genomes, and virions derived from these genomes will vary slightly from each other. In other words, it's a quasi-species. Likewise, a sample taken from a virus culture or an infected animal will contain numerous virions, many of which vary slightly. Consequently, a virus, a strain, a variant, or an isolate always refers to populations and not to single physical entities, and their descriptions are therefore based on average properties. Now let's consider some common types of virus mutations. The in vitro replication rate of pathogens is a common measure of fitness. A rapidly replicating virus in culture is interpreted as having greater fitness than slower growing strains. Correspondingly, the size of plaques, which are caused by necrosis or apoptosis upon infection of a cell monolayer, is a common proxy measure of viral fitness. Viral growth characteristics in cell culture, including plaque morphology, have been related to pathogenesis and disease severity for several human pathogens. For example, in West Nile virus, a small plaque phenotype showed less efficiency of replication in vitro culture compared to the wild type and was attenuated in its neuroinvasiveness. In the illustration on this slide, you see the plaque morphology of the parental yellow fever 17D vaccine virus, which is over on the left, and two variants. The small plaque sizes of the two variants resulted from a defect in their ability to spread from cell to cell rather than merely a diminished cytopathic effect. An Asian American lineage Zika virus strain formed two types of plaques, large and small. Growth kinetics of a large plaque clone was faster than that of a small plaque clone in viro cells. Genomic analysis of the plaque-forming clones obtained from the isolates indicated that the clones forming small plaques commonly had an adenine nucleotide at position 796 in the amino acid sequence, while clones forming large plaques had a guanine nucleotide at that same position, leading to a switch in amino acid from arginine to glutamine. Growth kinetics of a large plaque clone was faster than a small plaque clone in viro cells. These results indicate that the amino acid variation at position 230 in the viral polyprotein, which is located in the M protein sequence, is a molecular determinant for plaque morphology, growth property, and virulence in mice of Zika virus. During an outbreak of chikungunya virus in the southern part of Thailand in 2009, isolates from patient sera were found to be composed of a mixed population of small and large plaque phenotypes with respect to the plaque morphology in viro cells. So this shows that you can find a variety of phenotypes in patients and not just in the lab. Host range is a key property of a virus that reflects the diversity of species that it can infect naturally, and expansions in viral host ranges provides the potential for the emergence of a new disease. However, host range is often difficult to define, and many factors need to be incorporated, such as the host's susceptibility to infection, as well as the ability of the virus to undergo sustained transmission in the new host. The successful replication of a viral agent in a host is a complex process which consists of a number of interactions, most of them related to the coevolution of pathogen and host. Coevolution often leads to a species specificity of the virus and can make interspecies transmission difficult. Therefore, natural host range switches by viruses are rare events. However, when they do occur, the results can become severe because the viruses may then spread widely through non-previously adapted and therefore immunologically naive host populations. A conditionally lethal mutation is one that can replicate under some permissive conditions, but not under other restrictive or non-permissive conditions, while the parent wild-type strain is able to replicate under both conditions. In practice, the vast majority of conditionally lethal mutations are recessive, with loss of function of the gene product under the non-permissive condition. At the DNA level, conditional lethal mutations are often single base substitutions in protein-encoded genes that cause amino acid changes in the protein product. Single base substitutions in genes encoding RNA products are also known, and these often change the stability of stem loop structures. The molecular basis of conditional function in mutant proteins can result from the inability of the protein to fold or remain folded in an active three-dimensional structure under the non-permissive condition. Often the conditional phenotype of the mutant protein can be observed in vitro. Conditional phenotypes can also arise when a mutant protein prevents the assembly of an active protein complex under the non-permissive condition. Cellular proteases that recognize misfolded proteins often amplify the effect of the mutation and contribute to the conditional lethal phenotype. Now, a very common type of conditionally lethal mutation would be the temperature-sensitive or TS mutants. Here's an example of this. With Synbis virus, they found a number of different temperature-sensitive mutants. And remember that Synbis is an arthropod-borne virus, so it moves between an arthropod host and a vertebrate host. So they'll look at 28 degrees centigrade as a lower temperature and 39.5 degrees centigrade as the higher non-permissive temperature. Now, with TS6, you can see that there's substantial reduction in titer at the non-permissive temperature, the same as with TS20 and TS23. Now, it may seem like you're still getting a lot of virus being produced at the non-permissive temperature, but keep in mind that that's actually less than 1% of the virus that's produced at the permissive temperature. There's always going to be some virus production because these mutants tend to be leaky. Now here you can see what happens with TS20. This is TS20 at non-permissive temperatures, and you can see the nucleocapsids are attached to the internal surface of the plasma membrane, but they're unable to bud. Now if you take the same flask of cells with virus and move it to permissive temperature, you do a temperature shift, you get a change. So you shift the cells to permissive temperature, and within an hour, you start to see that virus has been released from the cells. So most likely a morphological change in a protein. Here's TS23 at non-permissive temperature, and you see that the nucleocapsids are randomly distributed in the cytoplasm of the cell. Here, when we shift to permissive temperature, budding commences. Now, there are other kinds of mutations as well. For instance, deletion mutations. These will occur spontaneously, particularly when the virus is passed at high multiplicity of infection, or MOI. If deletion is in a non-essential sequence, it won't be defective. These can be useful for mapping. Other kinds of mutations include drug resistance, virulence, pathogenesis, and so on. Keep in mind that these can often be linked. For instance, resistance to Tamiflu, a neuraminidase inhibitor used to treat flu, has been shown to be the result of a deletion. We'll talk more about deletions when we get into defective virus particles later on in this lecture. Co-infection, where multiple viruses infect a cell simultaneously, is important in the life cycle of RNA viruses. During co-infection, haploid virus genomes act in a manner similar to those of diploid organisms, exchanging genetic material randomly or preferentially. Reassortment of genome segments may create severely deleterious mutational combinations, thus speeding up the elimination of mutational load. Co-infection, however, may result in complementation, where viruses carrying different deleterious mutations may benefit from the normal products that each can produce, so that both types of viruses can be represented in the offspring. In contrast to recombination, complementation weakens the selection against deleterious mutations. In this way, it contributes to the stability of the whole virus population, as it maintains a high level of diversity without sacrificing the overall fitness of the population. So let's look more closely at complementation. Now here's an illustration of exactly how complementation occurs. So we're looking at two different viruses, the green and the pink, and you can see that their polymerase and their capsid proteins differ. So in the green virus, the capsid protein is defective. So when that virus infects a cell by itself, no progeny virus can be produced. Now with the pink virus, no polymerase is expressed, but the capsid protein is normal. But again, because there's no polymerase that can work, it's a mutated form, you don't get any progeny virus when that type of virus infects a cell by itself. But if you co-infect a cell with both of these, you can get progeny virus being produced, because one will produce a normal polymerase, the other will produce a normal capsid. And remember how viruses replicate. It's a process of assembly, not a division. So you get a mixture of viruses, and but you'll notice that the output virus will be like the parentals, because notice with this, with complementation, the genotype is not affected. Complementation is a sharing of gene products in a co-infected cell. It has its basis in the nature of virus replication and assembly. If both mutants are defective in the same gene product, then there is no complementation. In fact, you can use this to place mutants in functional groups. If the mutations complement, then they must be in separate genes. If the mutations fail to complement, they must be in the same gene. So here you can see how you can use complementation to do mapping of viruses. In this case, we're looking at a type of virus that has multiple genome segments. And we have Group A, Group B, Group C. You'll get no complementation between Group A viruses because they are all defective in gene 2. However, you can get complementation between viruses in groups A and B because different genes are involved. You don't need to know anything about the specific biochemical defects. Theoretically, there are as many complementation groups as genes. We can look at an example of this looking at our TS mutants in Synbis virus again. So here, if we take TS6 and TS23, remember that at non-permissive temperatures, the replication rate is going to be low. So when you grow your controls, you're going to get a low rate of infection. But if you do a co-infection, TS6 plus TS23 at non-permissive temperature, then you get a higher result, a higher production of virus than when either one was grown at non-permissive temperatures alone. And that was because of complementation. The complementation index is the ratio of yield of mixed virus growth to the sum of the yields of a single infection. So in this case, the complementation index is 290. Complementation is often the first genetic test carried out. It's easy and divides mutants into functional groups. It's the equivalent of chromosome mapping in a eukaryotic organism. Okay, so now we can move into recombination, and that actually involves the physical interaction of viral genomes. So your progeny viruses are going to be different than the parental viruses. And there are two types of recombination that we can see with viruses: intramolecular recombination and reassortment. Okay, so the first type of recombination that we're going to talk about is intramolecular recombination. Intramolecular recombination occurs in all DNA viruses that have been studied and in several RNA viruses. The nucleic acid has to break and reform. This is equivalent to crossing over in chromosomes. As an example in this illustration, you can see recombination that can occur with coronavirus. The result from a high frequency of recombination events in coronaviruses is the generation of novel viruses with a high genetic diversity, with unpredictable changes in virulence during human infections. With multiple species of coronaviruses circulating in the wild amongst different animal species that may constantly interact with one another, it is likely not a matter of if, but when the next recombinant coronavirus will emerge and cause another outbreak in the human population. This slide shows the mechanisms of replicative and non-replicative RNA recombination. The replicative mechanism is sometimes called copy choice or template switching. The replication complex pauses during the synthesis of the complementary strand of the RNA donor, in red, and dissociates from the RNA donor template. Then, the incomplete nascent RNA strand interacts with the acceptor RNA, in blue, where the replication complex reassembles and the synthesis of the complementary strand resumes. The complementary strand is indicated by lighter colors. The non-replicative mechanism is one of breakage and ligation. The two parental RNA molecules are degraded, and then the two fragments generated are covalently linked. And then we have reassortment. Virus reassortment, or simply reassortment, is a process of genetic recombination that is exclusive to segmented RNA viruses, in which co-infection of a host cell with multiple viruses may result in the shuffling of gene segments to generate progeny viruses with novel genome combinations. Reassortment has been observed in members of all segmented RNA virus families, but it is most prominently described for influenza viruses as a primary mechanism for interspecies transmission and the emergence of pandemic virus strains. For instance, reassortment accelerates the rate of acquisition of genetic markers that overcome adaptive host barriers faster than the slower process of incremental increase due to mutation alone. The emergence of new influenza genes in humans and their subsequent establishment to cause pandemics has been consistently linked with reassortment of novel and previously circulating viruses. With recombination by independent assortment, genes that reside on different pieces of nucleic acid are randomly assorted. This can result in the generation of viruses with new antigenic determinants and new host ranges. Development of viruses with new antigenic determinants through independent assortment is called antigenic shift. Here you can see reassortment of influenza virus. This method is used to generate the new strain of vaccine virus that's used each year. It's the HA and NA genes that are important for vaccine production. These are the glycoprotein antireceptors. This is what antibodies will recognize. So what you can see in this picture is that there's an attenuated donor master strain. They have to identify what they think the new virulent variant is going to be, and then they produce an attenuated vaccine strain that expresses the HA and the NA of the strain of interest, but is attenuated because of all the genes from reassortment that came from the attenuated donor. Okay, the next type of genetic interaction would be heterozygosity. Human immunodeficiency virus, like all retroviruses, is diploid. Each viral particle contains 2 RNA strands of positive polarity, each full length and potentially able to replicate. No other virus families, RNA or DNA, are diploid. Typically, both RNA strands in a retroviral particle derive from the same parent provirus. However, if an infected cell simultaneously harbors 2 different proviruses, one RNA transcript from each provirus can be encapsulated into a single heterozygous virion. When this virion subsequently infects a new cell, the reverse transcriptase may jump back and forth between the 2 RNA templates so that the newly synthesized retroviral DNA sequence is recombinant between that of the 2 parents. All subsequent progeny virions will be of this recombinant genotype. HIV strains with chimeric genomes thought to have arisen through homologous recombination have recently been discovered in nature. So heterozygosity, this is a term that you're familiar with in dealing with eukaryotic organisms that have a diploid genome. But normally we don't see this with viruses, only the retroviruses. However, we can use the term heterozygosity when you have a packaging error, an aberrant packaging, where you can end up with a multiploid particle. And you can even have 2 complete nucleocapsids within the same envelope. So packaging errors can also result in heterozygosity. Okay, and then we have pleiotropism. With pleiotropism, a single virus protein may have effects on several viral properties. Virulence is often affected. This is especially useful if an in vitro property can be tied to virulence in vivo. Then you can select for the attenuated strain without a lot of animal testing and just save that for the end of your studies. In this slide, we see an example of pleiotropism in Zika virus. The plaque size of the G796A mutant was significantly smaller than that of the parental strain. The mutation also reduced the growth rate of the parental virus in viro cells. We actually saw that in another slide earlier in the lecture. In addition, this mutation decreased the virulence of the strain in mice. These results indicate that the amino acid variation at position 230 in the viral polyprotein, which is located in the M protein sequence, is a molecular determinant for plaque morphology, growth property, and virulence in mice of Zika virus. Now we're going to talk about defective viral genomes. Some defective viral genomes are so damaged that they are completely lacking in biological function. However, there are 5 classes of defective viral genomes that are biologically active, and they can serve some pretty important biological roles. For instance, they may play a role in gene transfer and expression, they can regulate virus lethality, and they can play a role in evolution of host and virus. So we'll look at some of these examples as we go along. Many bacterial and animal viruses lie dormant in the infected cell, and their DNA may be integrated into the DNA of the host cell chromosome. The integrated viral DNA replicates as the cell genome replicates after cell division. The integrated viral DNA is duplicated and usually distributed equally to the 2 cells that result. The bacteria that carry the non-infective precursor phage, called the prophage, remain healthy and continue to grow until they are stimulated by some perturbing factor such as ultraviolet light. The prophage DNA is then excised from the bacterial chromosome, and the phage replicates, producing many progeny phages and lysing the host bacterial cell. Excision of the prophage DNA from the host chromosome DNA as an initial step in the synthesis of an infective lytic virus sometimes results in the removal of some of the host cell DNA, which is packaged into defective bacteriophages. Part of the bacteriophage DNA is removed and placed at the other end of the gene by the host bacterium. Such a virus particle is called a transducing phage because when it infects a bacterial cell, it can transmit the gene captured by the phage DNA into the next bacterial cell it infects. Transduction by bacteriophages is an efficient method for transferring the genetic information of one bacterial cell to another. And here we can look at this. This slide shows you mechanisms of mobile antibiotic resistance in bacteria, and that's something that we've all dealt with, the fact that bacteria can quickly develop antibiotic resistance. And there are different ways that they can do it. We're interested in the one that's circled in red on this slide, transduction. And in this case, what you have is bacteriophages accidentally package some of the genes from the antibiotic resistant bacterial cell, and then when that bacteriophage goes off and infects another cell, it injects that new gene into the bacterium and transfers the antibiotic resistance. But keep in mind that that bacteriophage that incorporated some of the bacterial cell's DNA is now defective because part of its normal viral DNA was replaced by the bacterial DNA of the host cell. So it's a defective virus, but it's playing an important role in the evolution of bacteria. Integration of a DNA copy of the viral genome into a host cell chromosome is an essential step in the retroviral replication cycle. Once integrated, the proviral DNA is replicated along with cellular DNA during cycles of cell division, as with any cellular gene. The provirus serves as a template for transcription of viral RNAs. Some viral RNAs are translated to yield the viral proteins, whereas a portion of the full-length viral RNA is recruited to serve as genomic RNA in progeny virions. By definition, a provirus is the genetic material or genome of a virus that is integrated into the DNA of an infected host cell. In HIV-infected patients on combination antiretroviral therapy, greater than 95% of the proviruses in the peripheral blood are defective. Previously, these defective proviruses were thought to be dead-end products with no real pathophysiological significance because they do not encode replication-competent viruses. However, it's been shown that these defective proviruses can produce viral proteins. So these proviruses are replication incompetent but are translationally active. These defective HIV-1 proviruses are therefore biologically significant because they have the potential to elicit an immune activation and may actually serve as a barrier to an HIV-1 cure. Oncogenic RNA tumor viruses are defective and may have a profound effect on the host. Cellular oncogenes have been incorporated into the virus genome, and then they can transform cells in culture and cause tumors in animals. Cellular oncogenes encode proteins that regulate the cell cycle. They induce the cell to enter S phase, in which DNA replication occurs. With infection by a sarcoma virus and integration of the provirus into the host genome, the expression of the oncogene results in transformation. Most transforming retroviruses are defective because the oncogene replaces part of the viral genome. In this figure, you can see that RSV is not defective because the oncogene is added at the end of the genome, leaving the GAG, Pro, Pol, and ONV genes intact. RSV and avian myeloblastosis viruses cause rapid transformation, while ALV does not. About 40 oncogenes have now been identified. They're referred to by a three-letter code, such as CIRC, often reflecting the virus from which they were first isolated. Some viruses can have more than one oncogene. Because they are viral oncogenes, we put a letter V in front of the three-letter name. The cellular homologs of viral oncogenes are called proto-oncogenes. To distinguish viral oncogenes from cellular proto-oncogenes, they're often referred to as V-onc and C-onc, respectively. Note that C-oncs are not identical to their corresponding V-oncs. It appears that the virus has picked up a cellular growth controlling or differentiation gene, and after the gene was acquired by the virus, it has been subject to mutation. Satellite viruses are also replication defective. They can only replicate in the presence of a helper virus. They require the gene products provided by this helper. It's interesting to note that often there's no homology or relation to the helper and the satellite. There's some important examples of satellite viruses. For instance, hepatitis delta agent is a satellite of hepatitis B virus. It can only replicate in cells that are also infected with hepatitis B. This can be the result of a simultaneous infection or a superinfection. But we do find that individuals who are infected by both hepatitis B and the hepatitis delta satellite have a more serious illness than those that are infected by HBV alone. Many plant viruses have satellites associated with them. For instance, TNV is a small icosahedral RNA genome virus that infects plant roots. TNVS is a satellite associated with many isolates of TNV in nature. This is an icosahedral T=1 RNA. The satellite RNA genome encodes only the protein that encapsulates its own RNA. All the functions required to replicate the RNA are provided by the helper TNV virus. Pseudovirions are virus particles with host nucleic acid instead of the viral genome. This is a potential mechanism for introducing foreign genes into animal cells. We've already talked about conditionally defective genomes. These are mutant genomes that are defective under certain circumstances, such as the temperature-sensitive mutants that we discussed, or host-range mutants. The next category, the DI particles, we're going to talk about in more detail. Defective interfering or DI particles are virus-like byproducts of infections that are unable to infect cells on their own because they carry mutations, typically large deletions, in essential viral genes. However, when DI particles and standard virus particles co-infect the same cell, the DI particles compete for resources that ultimately enable them to reproduce at the expense of the normal virus particles. A characteristic feature of DI particles is their emergence and outgrowth during high multiplicities of infection, or MOI, where numerous copies of the standard virus infect each host cell. Under such conditions, predator DI particles can productively interact with prey cells infected with standard virus. You can see that in the graph. As the MOI increases, the replication of virus is increasingly inhibited. In the figures at the bottom, you can see the structure of VSV and a DI particle of VSV genomic RNAs at the right, and EMs of the normal and DI virions on the left. Defective viral genomes can serve many important biological roles. For instance, gene transfer and expression. We've already talked about how they can play a role in transduction of bacteria and the transfer of antibiotic resistance between bacteria. In terms of lethality, it's possible that DI particles may arise late in infection of an animal and lead to attenuation of symptoms by reducing the yield of infectious virulent virus. With satellites, sometimes the lethality is increased. We talked about that with hepatitis delta. Constant selective force on virus to compete with defective virus can generate population diversity. Now let's look at some practical uses. Here's an example of using pseudovirions for gene transfer. The structural protein of many viruses have the ability to self-assemble into virus-like particles when expressed in an expression system. The major capsid protein of papillomavirus can self-assemble into VLPs, or virus-like particles, and have a receptor that recognizes many types of cells. VLPs were produced and purified by centrifugation, and in this illustration you can see that treatment with buffer can cause the capsimers to disassemble. You change the buffer and the capsimers will reassemble into capsids. This provides a mechanism to load the VLP with the foreign protein. And here you can see how that can work. Two different plasmids were used. Green lantern plasmid, so you look for the fluorescence, and PCMVB, you look for the expression of beta-galactosidase. 70% of cells infected by the VLP and plasmid were transfected. No transfected cells were observed when the cells were incubated with the DNA plasmid alone. And you'll notice on that slide it mentions lipofectamine. That's a drug that enhances the uptake of plasmids. So clearly, if you look across the top with the green lantern plasmid, you can see that plasmid alone, you really did not get expression. You didn't get transfection of the cells. When you used the drug, you got a little bit, but when you loaded your plasmid into the VLPs, you got a very high level of transfection. And you can see that same pattern when you use the other plasmid in the lower sets of pictures. Viruses like SARS-CoV-2 require high containment, such as BSL-3 and BSL-4 labs, and that can hinder the efforts at developing a vaccine. The pseudovirus system is a useful alternative approach that can effectively screen vaccines on pathogenic viruses outside of a BSL-3 or BSL-4 laboratory. One packaging system that has been used for the generation of SARS-CoV-2 pseudoviruses is the highly efficient lentivirus vector system. Viruses that are highly infectious and highly pathogenic, such as SARS, make it difficult to screen neutralizing antibodies, which is an important step in trying to produce a vaccine. However, the pseudovirus can only infect cells in a single round, has broad host range, high titer, and is not easily inactivated by serum complement. In the pseudovirus luciferase assay, the inhibition of viral entry into cells by neutralizing antibody is correlated to the decreased levels of luciferase signals in the cells. This method is superior to the conventional assay because of its simplicity, higher sensitivity and accuracy, suitability for high throughput experiments, and in addition, no live virus is used during the test. Therefore, this method could be used as an alternative for safely conducting serological studies in a rapid response in assessing the threat posed by SARS-CoV-2. There are researchers here at Tech who are using this lentivirus system to study SARS right now. DI particles can suppress the replication of standard viruses. The red arrows in this figure represent the inhibitory effects of defective interfering particles. DI particles can interfere with absorption, penetration, uncoating, replication, packaging, and/or release. With their shorter genomes, DI particles are able to replicate more copies per unit time and compete for resources with the standard viruses. DI particles have been shown to be able to activate immune responses, and other studies show that DI particles can be used as a vaccine against viral infection. Lastly, DI genomes display anti-tumor effects by inducing tumor cell apoptosis and promoting dendritic cell maturation. There are many practical aspects of virus genetics. When we consider virus evolution, we have to look at things like mutation rate, recombination and reassortment, antibodies that provide selective pressure, other kinds of host effects, and even drug treatments. Vaccine development depends on a knowledge of virus genetics, and of course is used to select for attenuated strains of virus. But lastly, I want to talk about viruses as vectors, in particular in the expression of foreign genes. Now, we did talk about using pseudoviruses as a vector, but let's look at gene therapy. Gene therapy is designed to introduce genetic material into cells to compensate for abnormal genes or to make beneficial protein. If a mutated gene causes a necessary protein to be faulty or missing, gene therapy may be able to introduce a normal copy of the gene to restore the function of the protein. A gene that's inserted directly into a cell usually does not function. Instead, a carrier called a vector is genetically engineered to deliver the gene. Certain viruses are often used as vectors because they can deliver the new gene by infecting the cell. Viruses are modified so that they're attenuated and they can't cause disease when they're used in people. Some types of viruses, such as retroviruses, integrate their genetic material, including the new gene, into a chromosome in the human cell. Other viruses, such as adenoviruses, introduce their DNA into the nucleus of the cell, but the DNA is not integrated into a chromosome. The vector can be injected directly into a specific tissue in the body, where it will be taken up by individual cells. Alternately, a sample of the patient's cells can be removed and exposed to the vector in a laboratory setting. The cells containing the vector are then returned to the patient. If the treatment is successful, the new gene delivered by the vector will make a functioning protein. Researchers must overcome many technical challenges before gene therapy will be a practical approach to treating disease. For example, scientists must find better ways to deliver genes and target them to particular cells. They must also ensure that new genes are precisely controlled by the body. There are a number of risks associated with gene therapy. This includes undesirable health effects, such as cancers and even death. If gene insertion occurs in the wrong location, it may affect the functioning of pre-existing genes that are vital within the genome. Virus vectors may infect healthy cells or tissues. The virus may revert to original form or mutate and become pathogenic. Virus entry may trigger an immune response, leading to inflammation, toxicity, and even organ failure. And the treatment needs to be repeated at regular intervals, increasing likelihood of adverse treatment response with time. So this is something that's actively being looked at, but it has a long way to go.